Assalamu alaikum and good afternoon and good morning. In fact, as the conference is going to be started in the morning. So first of all, I'm very thankful to the organizers who have given me this opportunity to present our piece of work in this conference, which is a prestigious one. So today, what I'm going to share with you is the antihypertensive and antioxidant activities of coumarin, a natural plant metabolite. So if we have a look on the non-communicable disorders, these are a high priority research theme at any institutions. These are becoming the public health emergency, and these are covering, and these are prevalent in more than 80% of the population, and out of those, 35 million deaths are occurring in developing countries. We, being the part of the developing countries, Pakistan, are facing a major threat for, for, for the expedited growth of these non-communicable disorders. Globally, if you look at the prevalence of the hypertension is globally prevalent amongst 1.39 billion people, and it has been estimated that by the year 2025, it will raise up to 1.6 billion. And in Pakistan, 26% of the population is forced to live with hypertension, and even the data is not known for how much people are living with hypertension without proper diagnosis and the treatment or optimized treatment. Hypertension also affects 18% of the population, of which the persons, 34% of these patients are having age more than 45 years. So keeping all these in considerations, the natural compound, the coumarins, have been focused of a current study, which is thought to be the fused benzene and pyrrole ring system. These are having a wide spectrum of bioactivities, including anti-tumor, anti-inflammatory, antiviral, and antibacterial. The presence of the coumarins in many plants have been found, and in the form of the different fruits, seeds, roots, and these are very common in some cassia species, in tonka beans, and many more plants. Why there is a need for the development of newer therapeutic agents? Don't we have different treatment modalities for the treatment of hypertension? Well, most of us might be knowing that there are multiple classes having multiple agents for the treatment or to combat hypertension. But even so forth, the cost of the therapy, prevalence of the adverse effects, patient non-compliance with the longer duration, these are the challenges which actually compelling us for the development of the more efficacious and relatively more safer agent for the treatment of hypertension. So in this current study, Coumarin has been developed to see its efficacy in alanine challenge hypertension. In this picture, the raw system actually is developed because of the challenge of the alanine, which causes pro-inflammatory cytokines, may lead to vasoconstriction, and ultimately leading to endothelial dysfunction, which is contributing in the development of the hypertension. So this study aims to provide the scientific plausibility for the use of this coumarin in hypertension based on its antioxidant activities. We also intend to evaluate its administration and its effect on ACE, messenger RNA expression of the ACE, NOS, COX2, and of kappa B in the experimental animals as well. The standard that we used, we subjected the standard for HPLC, and it has been found to have appreciable purity grade. When we dock this coumarin against ACE enzyme in the nitric oxide synthase, it has been found an appreciable energy with a positive influence that it might have having a binding capacity with angiotensin converting enzyme and also the nitric oxide, giving us a clue that it might have some positive impacts in hypertension, possibly involving ACE enzyme and nitric oxides. So this is a pretty cool system of micro, this a lab data acquisition system where we do a minor surgical procedure with anesthetized rats. We do cannulation, we do jugular vein cannulation for the dosing, and carotid artery has been cannulated to assess the pressure to determine the systolic, diastolic, and the mean blood pressure in these animals before and after the treatment. This is the PALAB system where in an invasive way and in a non-invasive way, we can also measure this and how the system works. We acclimatize rats in these chambers, and then through cocktail method, coupled with the PALAB data acquisition system, we do recording and see the effect of the test material on hypertension in non-invasive manner. So if you look at the effect on blood pressure, at week 3 and week 5, at both doses of Coumarin, both of these showed appreciable effect and showed blood pressure lowering activity. So giving us a clue that it might have having a good antihypertensive action, possibly mediated through multiple mechanisms. Then few of the analyses were subjected by using ELISA reader, by looking at the CGMP and NO levels. These were also found to be decreased with the treatment of Coumarin. Pro-inflammatory cytokines when assessed in the treatment, and diseased animals, along with the comparison with the positive control as well. These showed appreciable effects on the dose of 30 mg and the 70 mg per kg of these animals. When the antioxidant biomarkers were assessed, these were also found to be reduced appreciably at both tested doses. Finally, to look into the possible mechanism of action, the isolated tissue reactivity experiments were designed. Tissue preparations were isolated, mounted in the tissue organ bath assembly, and this system was coupled with the pressure transducer, isometric transducer, and the PALAB data acquisition system. Here we can see the myobath where the tissues were mounted to see the vascular reactivity before and after the treatment. And then finally, the effect on the messenger RNA expression of ACE, eNOS, and COX2 and NF-kappa B and messenger and NF-kappa B also showed positive impact of the treatment with Coumarin at both doses, 30 and 70 mg per kg. So by having this summary slide, which is showing that how this is mitigating the effect of ROS, how this is mitigating and antagonizing the generation of the ROS, and how the eNOS pathway is also antagonized by using Coumarin, and how the Coumarin is using to antagonize the detrimental effects of the alanine, actually provides a scientific base that it should have been used, and it is possibly involving these possible mechanisms for its efficacy. So to conclude with, Coumarin possesses antihypertensive activity in alanine challenged hypertension mediated through modulation of endothelium, enhancement of the NO and CGMP levels. It also showed protection against oxidative stress biomarkers, CAT and SOD, and upregulation of the messenger RNA expression of eNOS and ACE were observed, while the downregulation of the inflammatory indicators, the messenger RNA expression of COX2 and NF-kappa B was also observed. So keeping all these in consideration, it provides an evidence to its therapeutic potential for the treatment of hypertension and oxidative stress. So I I'm highly obliged and sincerely thanks to the chief organizer, Dr. Chief organizer and the team of Brain and Biomedicine. I also like to acknowledge the efforts of Dr. Bushra Shaukat, the Higher Education Commission of Pakistan, who has funded this project, and the Department of Pharmacology and Faculty of Pharmaceutical Sciences. Thank you very much. If any question, I would be happy to answer those.